dear students. It's my great pleasure and great honor to be here to talk about brain plasticity. Every one of us is different. The main differences comes from we have a different brain. So these different brain are created during our development. Basically, the whole life of our experience shape our brain. And today, my topic will cover the idea that the brain is changing all the time, and the basis of this change is now somewhat understood at the cellular level. And the ability to change our cognitive processing are also based on plasticity. So let me begin by saying that the plasticity of the nervous system, what is it? I can define the plasticity as follows. The ability of the nervous system to adopt a new functional or structural state in response to extrinsic and intrinsic factors. Extrinsic factors such as the experience you have based on the response to the environment. And this property of plasticity is in direct contrast to another property of the biological system called homeostasis. Most tissues in our body doesn't want to change. They want to maintain the functional state in response to various external stimuli in order to function properly. And the brain tissue also have this homeostatic property, but in addition, they have this very pronounced plasticity. That is, they will change. They will not only recover its original state in response to stimulus, but they will change to a new state that makes the brain different after the experience. Now, then the main forms of plasticity. Normally, we are capable of learning and memory because we can store the experience in our brain. Our brains change because of experience, because of learning. And that new state is normal plasticity. Many of the cognitive function comes from this ability. But there's an impaired plasticity due to many different processes, in particular developmental problems. The defects in the gene will cause developmental disorders in the brain that will cause mental retardation, schizophrenia, autism. All these brain diseases are developmental in origin. There are also adaptive plasticity. When the brain is subjected to injury, there's a change in order to adapt, to recover. And that change is the adaptive function of the brain that's endowed by evolution. And this injury will induce rewiring of your brain circuits so that you can function more normally. And this is a form of plasticity. There's also maladaptive plasticity, addiction, for example. Right? Drug addiction would cause changes in the circuits, in a different state, that you are craving for drugs. And that new state is a maladaptive state. Now, there probably is no evolution advantage to this property, but it's probably a byproduct because the brain is plastic. So there are situations where there is a plastic that's bad for you, and that's maladaptive. So all this plasticity is very important. Understanding plasticity in the laboratory will help us to understand what went wrong when our brain has a disease. Right? So this is a very important area in brain science, and brain disorders is now confronting our society in a major way. In fact, the WHO have calculated that the society burden caused by brain disease of all kinds are now among the highest, higher than those caused by cardiovascular disease or even by cancer. So this is an area of great importance. Now, let me first give you a very rough idea what the nervous system of the human looks like. We have a central nervous system which consists of the brain proper in our head. There's also a spinal cord. Those are called central nervous system. And these are the main seat of our cognitive function. There's also peripheral nervous system. The fibers sent out from the central nervous system to the periphery to control the muscle, control the organ, sympathetic and parasympathetic system that control the reaction in our body in response to a variety of stimulus. Now, what we are interested in most today is the brain, the brain of the humans. Now, the human brain is very prominent in its cortical structure. This tissue that surrounds the entire brain is called cerebral cortex. This is the most developed to a large extent in humans, much more than other animals. And the tissues within enveloped by the cerebral cortex tissue, the subcortical structure, are more ancient in evolution. Most of the mammalian systems have similar structure. But it's the cerebral cortex that makes us different, that makes us human. If you look at the evolution, the most prominent changes in the human brain in terms of size and the structure is the very pronounced cortical structure of the human, much more than our nearest relative, chimpanzee. Now, this structure, this homogeneous, looks like homogeneous cortical structure, actually are devoted to different functions. Right? The different regions of the cortex are responsible for handling information in different modalities: vision, somatosensory sensation on our body surface, motor that control your movement, and the language area. All these are localized to different brain regions. So one of the greatest principles that discovered over the past century is that different brain regions do different things. There's a functional localization in the brain. Now, if you look at this map of the brain, you can actually see if you do a section, biologists want to see what's going on in the brain, we cut a piece of the brain, a section across the brain, and look at the tissue. Then you'll find that in this cross-section, different parts of the motor cortex are actually also mapped into different regions of the body. For the top of the motor cortex are responsible for controlling our fingers and limb and faces and tongue. And this topographically ordered map is very important. For example, if you have this part of the region damaged, you'll have problem with your controlling your fingers, right? And it's also interesting that different part of the body, the brain devotes different resources to it. For controlling the finger and the face, large amount of brain motor cortex devoted to it, because this is too important. These are important regions. It's our hands, our fingers that allow the human has the ability to create tools. The face allows us to communicate. The social behavior in a large part depends on our facial expression. And so these regions are very important for the human, and it evolved much more pronounced into a much larger area. More tissue and more nerve cells are devoted to controlling this important region. Similarly, if you look at the sensation on the body, the somatosensory, the itch of pain, the pressure sensation on your body, those are also controlled by different part of the brain, of the somatosensory cortex. And this also have a map. Now this map, again, is devoted more to the fingers and faces and limbs and lips, which are most important, most sensitive area on our surface. And more neurons are devoted to sense the information coming from these areas with higher resolution. So these are the map. This is a distorted map. It's not in proportion to your body surface. It's actually distorted. And if you plot the actual shape of the areas that are devoted to different regions, you'll find that this is a distorted human being. They're small human being with different sizes. This is called the homunculus in your brain. Now, this map are very much similar among people. We all have similar organization. But this map is not static, it can change. Now, there's a very good example in humans. If you take this is data from an orchestra in France. People in the orchestra were taken to the brain machine scanner to look at what the area in our brain that are mostly devoted to the playing, to controlling their left fingers. The string players use their left fingers a lot, and that constant practice from a young age makes their brain area, the motor cortex in the right-hand side, that are controlling the left fingers much higher, much larger area. Now, string players compared to control, which are for those other musicians who doesn't play strings, they are control, and the area is much bigger than the control. And interestingly, if you started practice early, the area is larger. Even if you're a teenager, you start to play strings, violins, viola, or cello, you will have a larger area in your motor cortex devoted to that controlling your left fingers. Right? So this is a clear example of plasticity in your brain that can occur early in development and throughout your life. This is plasticity at a macroscopic level in your brain. But in a microscopic level, if you look at the cells within the cortex, you can do biological tissue staining. If you stain the cell, all the cell bodies, you see the cortex is packed with cells, millions and millions of cells in a small, tiny cubic centimeters. And these cells, you don't see much structure if you do a staining called Nissl staining. But if you do a different staining, in microscope, if you don't stain the tissue, it's all grayish, you cannot see the structure. If you stain with a stain called Golgi stain, which reveals the entire structure of the cell, you'll see that the cell body is only part of this nerve cell. In fact, each nerve cell has extensive processes that extend to form a very complicated network. Now, in this staining, only a few or less than 1% of cells in the cortex are stained. You already see this very complicated structure. Right? So if you have every cell stained, you would be totally black. You couldn't see anything. So this is a very complicated structure. Now, this structure consists of, in the human brain, 100 billion cells in the human brain, nerve cell, we call neurons. And they make connections. Their processes make connections with each other. Each cell makes 10 to the third, 1,000 connections or more. So you have a 10 to the 14th connection site called synapse. Now this forms this complete neural network in the brain. Now these nerve cells are not identical. They have a different shape and different morphology, different function, different physiological function. So there are hundreds actually different cell types that form this network. Among these 100 billion cells, you have hundreds, even thousands of different cell types. Exactly how many cell types in our brain is still unknown. In fact, one of the hard area in neuroscience to figure out at a single cell level how many different types are there in our brain. So this is still a quite open area. Let me look at let's look at a transparent brain. Right? There's a brain tissue, 1 millimeter mouse cortex, mouse brain, transparent by treatment, and then look at the fluorescent staining of the structure within the cell, within the tissue. And you can see this process in each neurons send out the long-run processes. Some of the processes actually are clearly visible, and this is only a few % of the cell that are labeled. And in fact, figuring out how the nerve cells connect to each other is a prerequisite to understand the structure of the whole brain. And so there is the so-called, after the Human Genome Project, the sequence of complete DNA structure of the human, the next big science project that people are facing is to characterize every connections from every neurons in the brain. It's called connectome. Connectome following genome is one of the biggest big science projects that many scientists are working on. And we have studied this in China with reconstruction, three-dimensional reconstruction of the neurons. In this particular example made by two colleagues in China, they plot the axon projection, the process of only 52 cortical cells, cortical neurons. Only 52 cells are plotted, each color with a different color. You see that these 15 neurons, only 15 neurons out of billions, already made this complicated structure in the mouse brain. So we can see that how these structures so complicated to figure out how they work is really a big challenge. Now, within these complete networks, there are many neural circuits. We call neural circuits because these are the connections that really carry out specific functions. These connections are sent out by a process called axon and dendrites. And these circuits form locally form so-called local circuits. They also form long-range circuits from one region to another region, like what I showed in the previous slide. Each neuron can make projection to a very long distance across the entire brain. We see this circuit. Let me give you an illustration. I think many of you in the audience probably doesn't know much of neuroscience. If you had an injury, an accident, you go to the doctor, you want to see whether you have damage in your nervous system. The doctor, the first thing they will do is to ask you to sit on a chair using a hammer to tap your knee and see if one tap causes your feet to lift up. Right? This is your knee-jerk response. This is carried out by a very specific circuit, neural circuits in the brain. In fact, the neurons responsible is in the central nervous system, in the spinal cord. The spinal cord neuron and the peripheral neurons called sensory neurons that sends out a process to your muscle. When you tap your knee, the sensors in the terminal of these nerve cells will send a signal to the spinal cord. And in that, they make a connection to two types of cells. One cell is called motor neuron. That motor neuron sends out a long process going back to the same muscle to cause its contraction. So once the physician tap your knee, your feet will lift up. That lifting up process is caused by contraction of the muscle on top of your leg. But on the same time, the muscle on the opposite side, beneath your leg, should relax so that the leg can lift up. Right? So this opposite action on the so-called antagonistic muscle is caused by the same signal that comes from the spinal cord, but it sends information to an inhibitory neuron, an interneuron. There's a local processing, convert the signal from positive to negative. So that signal inhibits the nerve cell that innervate the opposite side of your leg, so cause it to relax. So the top of the muscle contract, lower muscle relax, so you have a knee-jerk reflex. Right? So this is a very simple, probably the simplest neural circuit that worked out, right, by Charles Sherrington in Cambridge at the beginning of last century. Right? This is the first neural circuit. Now this is a local circuit, controlled within the spinal cord. But there's a long-range circuit. I mean, you can think that the physician asks you to relax on the chair so that I can hit your knee. But if you refuse to let the leg lift up, you can do it. You can control your leg and refuse, and the leg will not respond automatically. And that control signal comes from the brain. So the brain can send a long-range signal to control your leg muscle so that you would not undergo the reflex. So this is a long-range circuit. So this is the type of circuit that controls not only simple reflex, but even more complicated cognitive function. We see an object, we react to it, and that's a similar processing, but the process is much more complicated within your brain circuit. The sensory input comes in. After processing, you have a reaction of through your muscle. So that's many of the cognitive process. Now, a typical neuron consists of dendrites. Is a process, a tree-like process on the cell body. There's also each neuron sends a very long axon that can communicate information to other cells. Right? So long-range connections are made by this long-range, very long axon. Some of the axons are 1 meter long, from my spinal cord to my tip of my finger. That axon is very long. Right? So this is the typical neuron. Now there's a variation in the type of structure in the morphology and physiology, but most cells would have the dendrite and axon. And the way that this information is conveyed is by electrical activity, the so-called action potential, or nerve impulse, or called a spike. It's a small pulse of electrical activity that the cell, if you put an electrode into the cell, you'll find that the cell has a negative potential voltage inside the cell. And when the stimulus comes, the cell membrane becomes less polarized. The potential drops. When it drops to a level called threshold, then there's an explosive depolarization of the potential change in the membrane. And the 1 millisecond duration depolarization of the membrane, that's the signal. And this is the unit for communication. The communication by pulses, by these spikes or action potentials, by a code that is not dependent on amplitude of the spike, but on the frequency of spike. The way that the stimulus strength, the information amount is coded by the number of spikes, the frequency of spike. The spike, each spike will have the similar sizes, but they have a higher frequency where you have a higher stimulus. Right? So this is the coding of the nervous system. Let's take a look with a movie showing the process of this synaptic transformation. In the human brain, there are a lot of blood vessels. These red things are blood vessels providing energy, but within them there are neurons. This is a nerve cell. The neurons have these long axons, single axon that extends to a long distance. There's electrical activity conducting the signal. The activity is not electron, it's ion. The ion moves from outside into inside. Positive ion goes in, makes the negative potential inside the cell depolarize, and produce a wave. That wave pushes ahead to the end of the fiber. That's the transmission process. When the signal reaches the end of the axon, it doesn't go directly to the other cell, it goes to a gap. That gap is called synapse. And the way that the information is transferred is through mediation by chemicals that are released by the presynaptic or the neurons. That chemical release opens up channels, ion channels in the next cell, and that channels allows the ion to flow into the cells and create another wave of electrical activity in the next cell. So the activity, the electrical activity does not transmit it directly, it's mediated by the chemicals. And then there's a nice thing about this synaptic transmission by chemicals, because the amount of chemical you can release and the type of chemical you release could be changed. So different synapses could release different chemicals. Some chemicals would open up pores in the postsynaptic cells, allowing positive ion to flow in. Then the next cell will be excited. Because the positive ion flow in, it depolarizes the negative potential. But it can also, in some other synapses, the chemicals, a different chemical called transmitter, opens up the pores that allows only negative ions to get in. And then the signal will be making the inside of the cell in the postsynaptic cell more negative, and that depolarize cell. So we can convert a signal from positive to negative through a inhibitory synapse, we call it. Right? So this synapse is very important because this is complicated structure of the synapse, allow it to change under various type of conditions. For example, repetitive use of the synapse will change the property, change the efficiency for its transmission. And you can have most of our neuroactive drugs works on synapse because synaptic structure is most sensitive to drug, sensitive to changes from the environment. So plasticity of the brain mostly is controlled by the activity. This activity is electrical activity. As I said, this is an ion-based activity, not like the activity in our cables, which is the electron. Right? This ion-based activity propagates slower, much slower than the electron in the cable. It propagates at the speed of the sound, right? hundreds of meters per second. Now this activity will occur always when you have sensory input, you carry out a motor action. Any cognitive experience involves a lot of activity in your brain. And this activity comes in, it changes the structure and function of the neurons and synapse, in particular the synapse. Synapse is the most sensitive area for change by activity. So our focus is on synapse. Once this structure is changed, then you have the phenomenon so-called learning and memory. So what is learning and memory? Learning and memory is simply a process where you create a lot of activity in your brain, and that activity changes the synapse of a particular circuit and leave an imprint in this circuit so that circuit is changed forever. And next time the experience comes, you remember something. Right? So any memory is stored in a form at a synaptic structure and functional change. And once that plasticity occurs, then you have learning memory, you have change in your cognitive process and behavior. So the question has been for almost a century. That is, we know that activity is doing a lot of things to the brain, but how? How does the activity change the synapse? That's the question. And in the last couple of decades, this area has made tremendous progress. We now know how activity modifies the synapse. Right? The work started from a Canadian psychologist who proposed a very interesting hypothesis nearly 60 years ago, Donald Hebb. He proposed that how does activity change the synapse? Probably by correlation. Because when you have an experience, one cell triggering some cell in your brain, that cell would also activate some cells that it's connected to. And when the experience constantly occurs, repetitive occurring, then this activation of these two cells will be correlated. They will synchronize the fire for repetitively. And if that correlation of firing changes the synapse, then you can store something, connections. So for example, you have a presynaptic cell has this firing pattern. Each one of these is the action potential that codes the information. The postsynaptic cell happens to also fire that in response to that. And then the connection between them can be increased. This is what Hebb's postulated. It's called the synaptic strengthening or stabilization by correlated activity. And then a molecular biologist, Gunther Stan, interestingly, proposed in the 1970s that we need an opposite change as well. Now, if everything is changing all the time, then pretty much after a long time, all the synapses will be saturated, will become potentiated. So you need a process of tuning it down. What's going up may also go down. Right? So this is the hypothesis that Stan proposed is that the activity is not correlated. For example, these cells firing, but the other cell, another cell is connected to are not firing, probably receiving other inputs that inhibits its firing, then the synapse between them should be weakened. Right? So this will be a complete set of hypotheses to explain how activity modify your circuits. And this rule, HAPS learning rule, is called, can be summarized in one sentence, that cells that fire together wire together. Right? This is a very famous statement in the area of plasticity. Right? Remember this is one message number one, cell fires together, wire together. Now, the evidence for this, after HAPS proposal, comes up a few decades later. In the 70s and 80s, people start to find that if you stimulate an input of a neuron repetitively with high frequency, like 100 hertz stimulation, and the response, the synaptic, the synapse made by this input will be increased. Their efficiency will be increased. There will be potential. The synaptic efficiency or efficacy is measured by electrical recording methods, and that amplitude of that response show that the response is increased persistently for a long time. So it's called long-term potentiation. And if you fire with a low frequency, the synapse become reduced, efficiency become reduced. It's called long-term depression. Now, HAPS postulate can explain this very easily. HAPS says if you fire high frequency, the postsynaptic cell is likely to be fired because you pass the threshold to fire postsynaptic cell. So presynaptic cell firing high frequency, postsynaptic cell also active. So you have a potentiation. Now, if you fire at a low frequency, the response in the postsynaptic cell are not accumulative. You cannot accumulate above the threshold, so it does not fire. So you have presynaptic cell fire, postsynaptic cell not fire, so it's an uncorrelated activity, so you have a depression. So this explains the HAPS learning rule. Now this synapse that change is happening at a very simple, interesting structure called a spine. The dendrite where you receive the input from other axons actually has protrusions on it. These protrusions called dendritic spikes are the site where the presynaptic cell, the axon terminals from the other cells will end and make communication. Right? So there's this very strange structure that's responsible for synapse. So why one needs this structure? Why not just send information directly to the dendrite? Well, the idea is that having this structure allows the synaptic transmission efficiency to be controlled. If you have a very thin spine, right, a very thin neck, the signal will be hard to be communicated to the dendrite. So the signal definitely reduces much faster. Right? So by controlling the shape of the spine, you can control the efficiency of transmission. This has been demonstrated. We're not showing original data. This is a cartoon that depicts the axon terminal coming from other cells that make a bouton, a swelling on these dendritic spines. This is where all the transmission goes on. And these spines are highly dynamic. If you look with time, they're actually changing with time. Right? This is a dynamic structure. When you have a so-called LTP, long-term potentiation, that this connection becomes strengthened, the spine could be enlarged. When you have an LTD, long-term depression, the spine can shrink, sometimes even disappear. So by changing the size, the shape of the spines, you control the synaptic structure, in turn control the efficiency of transmission. Now we can actually see this changing in real life by the current technology called two-photon imaging. You can open a window on the brain, on the mouse brain, use a microscopic technique called two-photon imaging. You can look at the cells within the cortex. You can look at the presynaptic axon comes in, forming this swelling, the bouton that ends on the protrusions, the dendrites protrusions on the dendrite called spines. And if I even look through different dates, looking for the same dendrite, same spine to see whether they change. This technology, which is now available for about 10 years, allow people to actually look what happened during learning and memory. Right? So for example, in our recent work, we show that when you produce this long-term field memory, right? So you pair a sound with a foot shot to the mouse. Right? You constantly pair these two. After a while, the mouse, whenever the sounds appear, the mouse would freeze in anticipation of the shot would come in. So that's called a long-term field memory. This field memory can last for many, many days. It's a very robust form of field. But when the field is generated, you look at auditory cortex, you'll find that there are new synapse formed after the field memory occurs. Now this is before the field memory is produced. You have a presynaptic bouton ending on a spine, which is labeled fluorescent red. The green and red point in this yellow region is the synapse. Now this synapse will increase more efficient because after the field memory is created, a new spine will appear to make the synapse with the same bouton. So this bouton becomes more efficient in transmission. Well, this is one way. The other way, you will see that when here's a spine which has receiving information from a bouton before field memory is created. And after field memory is generated, you see another bouton is formed on the same axon. So making the transmission much more efficient. So what we show is that in fact there's an increased formation of this new synapse that after the field memory is created, and this increase in the formation of the bouton and the spine produce new connections, making the efficient that new connection is stable, and that's where the long-term memory is stored. And this is the evidence that not only you can change the shape of the synaptic structure, you can also change the number of synaptic structure. You make new connections as a form of learning memory. So Habs proposed this idea of learning rule to explain how we can remember the experience. The correlated activity, he said, would strengthen connections. Now you strengthen the connection among a group of neurons to store this memory. And that storage is at a connection, at a synaptic connection. Now what does it mean by recall the memory? Hab says that recalling of the memory is just to reactivate the set of cells that you have, make them link together by experience. So a specific assembly of cells created by experience, reactivating those assembly is the recall of the memory. Now we can see how this works. Let's suppose we have a circle. We don't remember a circle. Now each segment of circle will activate a particular neurons in your visual cortex. Now when a circle repeatedly appears, this group of cells will repeatedly fire in response to this circle. Now after a while, repetitive firing cause long-term potentiation of synapse. The connection between this group of cells will be strengthened. So you have a set of neurons defined by your circle. And that's the memory of the circle, because if you come up with a partial stimulus that activates a part of this assembly, you can reactivate the whole assembly because they are more strongly connected. Right? So this is how you recall the memory. Now you can extract it. If you see how you remember an object, not a circle. The object has many features. It has a motion, it can have a color, have different context, have different contrast. Each one of these is coded by a set of neurons that assemble, coding for that particular feature. All these sets activate together when the object appears in your view, in your field view. And this repetitive appearance of this object would set up this super assembly of neurons in your brain. And that gives you the memory of the object, right? You memorize your grandmother's face. Right? The grandmother's face has different, there are a set of cells coding for the grandmother's hair, nose, eye, different part of the grandmother's face, features of grandmother's face. Each one is a large group of neurons that were linked together. Now, the grandmother's appearance in your childhood, during your childhood, constantly set up correlated activity among this set of neurons, creating a super assembly of a grandmother's face. So you can remember a grandmother's face in your brain by this super assembly of cells. So the formation of this cell assembly idea is very powerful, and probably the most reasonable hypothesis to understand how we can remember a concept, code a concept, right? For example, the grandmother is a concept. Grandmother's concept consists of her face, right, which is coded by super assembly, as we said. But we also have assembly code for her, the sound he has sung to us, the stories she has told, her name, her smell, everything. That entire set of things related to a grandmother are stored in super assemblies everywhere in your brain, distributed in different regions. So the grandmother's concept is formed by setting this assembly. So when you are now seeing your grandmother's name, you immediately trigger the super assembly. You can remember her face, her story she told. This all within a huge assembly. So we call this assembly, the entire assembly, the concept storage site. It's not a single cell that codes your grandmother. It's a large group of cells that codes for grandmother, every aspect of grandmother. Now this is certainly a hypothesis, and it is being actively being tested, that the component of the sub-assembly are scattered in different brain regions. And the strengthening of long-range connection between this is the way the concept is formed, and the partial activation of this assembly leads to the idea of a concept, evoking the concept. So there's some evidence for this. For example, now we can scan the human brain by non-invasive method, and many hospitals have this facility. It's called brain scanner. You go to the brain scanner, they can tell you which part of your brain is active when you're doing something. Right? In this positron emission tomography, the so-called PET imaging, it shows the region that the glucose are highly used when reflecting activity is high. So you just flashes a few words, flashes the grandmother, the words onto your visual, your eye. You will see that the scanner would show activity in the back of your brain. That's called visual cortex. Rapid flashing, the reception site for visual information. If you just produce sound, grandmother sound to the eye, to the ear, to the subject, the auditory cortex are activated. If you ask the subject to say grandmother in the machine to produce language, the language area will be active. Right? So this is all we know, that different brain regions are coding for different functions. But however, if you ask the subject, close the eye, don't do nothing, but think about your grandmother. Just the concept of your grandmother, right? What happened to your brain is everywhere right now, right? So that suggests that the concept of grandmother or anything associated with grandmother are stored all over the place in your brain. That activity is hard to understand, and it's the goal of neuroscience, brain science, to figure out what's going on. Right? This is very amazing. But all these complicated structures are formed during development, after birth. Now, if you take a human brain, 1 month after birth, look at their networks. It's very few. There are a lot of cells. All the neurons are born after birth, right? But the connections are made after birth, 2 months, 2 years. The first 2 years, there's a tremendous growth of the process, the formation of the network. And that's amazing, because this network is responsible for all the functions of the human body. It's not there when you are born. It's formed after birth. Now, in fact, if you look at the spine, where we said it's the site where the synapse is made, there's a tremendous growth during the first few years, spine formation, but soon there will be a spine pruning. The number of synapses are dropping. The connection becomes simpler than what you had early in life. And that gradual pruning is a very important process because it leaves the connection that's useful, that's proper, and get rid of those connections which are not useful, which produce non-productive function. Like little kids, baby that hasn't started to walk, they're crawling on the floor, they couldn't coordinate themselves very well. They're on the rising phase, they're forming all of networks. When they start to walk properly, they have pruned their circuits to be more proper for their motor action. So they start to have a normal function. Right? This synaptic pruning goes on throughout life. In fact, the spine gradually decreases with aging. So normally there's an aging process. Now the spine, the synapse gets lower and lower, the formation of synapse, the ability declines. That's accompanying for all aging. But if you have genetic defects, the pruning process is altered. You may have autism or schizophrenia because the circuit you form are different. Right? So that's how the neural circuits develop. There are critical periods during the early life that you form the circuits. The neural network undergoes substantial pruning in response to activity. You've got to have activity in order to modify the synapse, right? to change the synapse. Different brain circuits have a different critical period. For example, in vision, if you have an imbalanced input in your eye, in a small baby, it needs to be corrected quickly, like cataract, right? You have to do cataract surgery early in their life, otherwise the cataract blocks your vision, proper vision. You pass the critical period after 3 years old. The vision circuit is already established, it's hard to change. Right? So the language also, you have to learn the language early in life before 7 years old. Otherwise, it will be very difficult to learn language, any language. Right? This is a very important critical period. Now the structure, in other words, we are carried out in our brain. Everybody has a different structure. That structure is formed during development, during the critical period, as well as after the critical period, because the pruning and the refinement of circuits still goes on, even to a lower extent in adult life. So we are different because our neural circuits are different. And this is and that neural circuit is the changes of neural circuits caused by learning and memory. So basically, our neural circuits has the imprint, has a learning and memory imprints through our lives. All our earlier lives are imprinted in our structure by changing the synapses. So development plasticity is high, but adult plasticity is also still there because in adult we can still learn. Large-scale rewiring is possible in early life, but in adult life, as I show you, you can add a few synapses. You have an existing synapse that you make it more efficient. You change the function, you change the structure, and those are limited circuit rewiring still going on in adult brain, but much more limited. Now, so there's a time where this adult plasticity are awakened. Once the injury occurs, there's a response to form new circuits. Right? The evolution and adult regeneration is response only after the injury. So you can repair the circuits. Right? Then there's a large scale change. There's also the reactivation of the adult brain. You can do various manipulation in animal models to show that the developmental plasticity can be reawakened. Now, I want to ask the most important question that everybody is interested in: origin of intelligence. Which is more important, nature or nurture, genes or environment? This is debated all over the place, all the time. Now, according to what I'm saying, that brain function is defined by the structure of the neural network, which is in turn shaped by the postnatal experience induced by activity induced by experience. So the circuit that determines the intelligence is actually shaped after birth. Right? It's not endowed in the immediate gene level, but the inherited gene is important because it's necessary to produce the building blocks for the neural network. And the formation of this network requires input from the environment. So in other words, genes offer the construction material for your network. It's the experience, the environment that builds the network. So now, which one is more important? 50-50? I think the question about which one is important is the wrong question. You cannot ask that question because both genes and environments are 100% required, 100% important. You cannot have either one go wrong, right? So this is how we should view intelligence. Now finally, a set of experiments that tells you the plasticity of cognition process. Now we all know that self-awareness can be reflected by whether you can recognize yourself in the mirror. A baby can recognize themselves in the mirror by their 2-year-old. By a 2-year-old, 65% of baby can look at a mirror and know that is themselves. Now, how do you show that? You secretly put some mark on the face of the baby, or a monkey, or other animals. You give the mirror and you see what they do. And the baby look at the mirror with a scratch the mark on the face. So that they are aware that face in the mirror are themselves. So these human and great apes, in fact, have passed this mark test. They are capable of recognizing themselves, but not monkey. Monkey, despite 50 years of work, have not been able to successful to demonstrate that they can recognize themselves in the mirror. Now, human baby can do that by 2 years old. Psychiatric patients and autistic patients sometimes fail to recognize themselves. They show no interest in mirror. They cannot pass the face mark test. So are they lacking in their self-awareness? It's an important question to answer. So recently, 2 colleagues of mine have done an experiment to train monkey to recognize themselves in the mirror. Right? This is an experiment done by these 2 colleagues. You put a laser pointer on the face of the monkey. It's slightly higher power laser that produces an itching feeling. Right? So the monkey would, in response to this sensation on the face in the mirror, monkey is sitting in front of a mirror, they will scratch. So they make association of an image in the mirror with the sensation of their own body. After this training of association for a few weeks, then they suddenly realize that animals in the mirror represent themselves. Right? This is one way of training. There's another way of training we call the visual proprioceptive training. Not involving the face now. You put a spot of light on this on the side of boards on the side. Ask monkey to touch that. The monkey is sitting here. They can only see that spot through the mirror because their head is fixed. Right? So if they touch the point right, they get the food, they get a reward. Right? They will do this training. In the beginning, they couldn't do it, but after many weeks of training, they can do very well, like this in late phase of training. They can do this touching very precisely, very quickly. So what is required for monkey to do this? Monkey has to recognize the image in the mirror and their own body. Where is their own body? Right? So they constantly associate this. They will be able to, finally they will realize that the image in the mirror are themselves. So they can pass the face mark test. This mark was put on by monkeys is not conscious, and then they would look at the mirror and they touch their face. This is a standard face mark test. Now, once they have learned this, they show a lot of spontaneous activity. They pull hairs on their face. Right? This monkey with the green collar was trained. Like start to look places where they couldn't see before. In interesting places. Now the other monkey, the yellow color monkey was an untrained monkey who was sitting there, who was really in the same cage for half a year. Really, he couldn't learn. Was not trained for this association. He never passed the test. So even these cognitive behaviors can be trained. In monkey, you can train it. So then you can study circuit mechanism. What is responsible for this ability, the emergence of this self-awareness? And a human baby can acquire this probably through learning. Right? So this is our conclusion. In fact, in the past 6 months, experiment done in colleagues in France have shown that you can speed up human baby, 1 year baby, after training of association would recognize himself in the mirror rapidly. Right? So that means that the learning ability, learning actually produces the cognitive ability. And the impairment of this awareness in autistic or psychiatric patient might be treated by training. Okay, so I should stop here and thank you for your attention.
Yeah.